May-June 2019, variant 2-2, and this is for A levels. My name is Miss Waheeda Ahmad. I am from Bagh school and I will explain question number 6, that is about enzyme and some investigation. Now see what is question number 6. Yes, question number 6, it is catalase is an enzyme that catalyzes the breakdown of hydrogen peroxide, which is a waste product of cell metabolism. The reaction catalyzed by catalase is shown in figure 6.1. You can see the reaction. Hydrogen peroxide will break down into water and oxygen by the enzyme catalase. This is figure 6.1. A student carried out 2 experiments to investigate the progress of the reaction shown in figure 6.1. Potato tissue was used as the source of the enzyme. 6 pieces of potato were cut and each measuring 20 mm into 10 mm into 10 mm. In the first experiment, hydrogen peroxide solution was added to 3 of the pieces of potato tissue and the progress of the reaction was measured. Figure 6.2 shows how the first experiment was set up. You can see there is a syringe in which hydrogen peroxide is filled. There is a flask which is fitted with a cork with a hole, and from one side the hydrogen peroxide will be poured into the flask, and there is another hole in the cork through which a delivery tube it is given, and it is inside a cylinder, and below there is a beaker containing water, and inside potato tissues are present. Question is, suggest how the progress of the reaction could be measured. During this reaction, when the hydrogen peroxide will come into the flask, come in contact with the potato tissues. Potato tissues are having catalase enzymes. Breakdown of hydrogen peroxide will start as soon as they come in contact with the potato. As a result, oxygen gas will produce. Through the other tube, the oxygen will go into the beaker, which is everted into water. The level of the water will go down when this cylinder will start to fill with oxygen gas. So level of water which will become lower can be measured, and the volume of the oxygen collected inside the cylinder in centimeter cubes can be measured using these apparatus. So oxygen will go into the beaker which was inside water, everted in water. There was a certain level of water inside the beaker. When the gas will go into the beaker and it will fill it, as a result the water will go down, the level of water will change, it will go down, it will decrease, and volume of the gas collected, that is oxygen in centimeter cubes, can be calculated. This is the answer. Second part of this question, in the second experiment, the student cut each of the 3 remaining pieces of potato to obtain 6 pieces, each measuring 10 mm into 10 mm into 10 mm. Using exactly the same condition, the student measured the progress of the reaction and obtained different results to the first experiment. Explain why the result of the second experiment were different from the results of the first experiment. In the first experiment, if you see, the size of the potato tissue is larger, it is bigger, 20 into 10 into 10. But in the second investigation, the size of the potato tissue is smaller, that means more surface area. In the second experiment, there is greater surface area of the potato tissues. When hydrogen peroxide will come in contact, so more catalase molecules will come in contact with the hydrogen peroxides because more cells are exposed while cutting because of larger surface area. Larger number of enzyme molecules and active sites are involved. They will make larger number of enzyme substrate complexes. As a result, faster reaction will be there. As a result, more product will be made, that is oxygen, and volume of the oxygen collected will be more. So what is the reason? Larger surface area because tissue is smaller. More active sites of the enzyme, they are exposed because of the larger surface area. More active site mean more enzyme substrate complexes, more faster reactions. As a result, the product will be more, that is volume of the oxygen collected will be more. The student then B part of the question investigated the effect of temperature on the activity of catalase. On figure 6.3 sketch a curve to show how temperature affects the activity of enzymes such as catalase. If you remember in the book there is a graph given for the effect of temperature on the enzyme activity. That is from the zero steadily the reaction will start to increase and almost near 60 it will steeply go down. So this will be the line on the graph. So this is the end of question number 6.